Okay, thanks everybody and welcome. Sorry we're starting a couple of minutes late. Today, my name's Elise Pendall. I'm the president of the AGU Biogeosciences section, and we're really happy to be here this afternoon. We've had such a great week. Our centennial sessions have been going really well, and yeah, it's been fairly busy. We're probably all a little bit tired. I know I am, but I'm really excited for this afternoon's speaker. Today, I'm pleased to introduce Dr. Colleen Hansel. She's this year's winner of the William S. and Carelyn Y. Reeburgh Lecture Award. It's co-sponsored by the Biogeosciences and Ocean Sciences sections. The Reeburgh Lecture that we'll hear this afternoon is given by a scientist who embodies Reeburgh's tradition of applying novel instruments to illuminate key issues in global biogeochemistry and marine geochemistry. Dr. Hansel's research has been called somewhat hard to pigeonhole in traditional disciplines. She was trained as a geologist and a soil scientist, and she's grown into a visionary marine biogeochemist with expertise in analytical instrumentation that bridges the fields of mineralogy, biogeochemistry, and microbiology. She completed her PhD in soil biogeochemistry from Stanford, received tenure at Harvard, and then moved to Woods Hole Oceanographic Institution. She's recently been a fellow at the Max Planck Institute for Marine Microbiology in Bremen, Germany. Please join me in congratulating and welcoming Colleen.
Thank you. Thank you for the introduction, and thank you all for coming. I know it's Thursday afternoon, everybody's getting a little worn out. And it really, truly is an honor to be up here and to be able to give a lecture in honor of Carelyn and Bill Reeburgh. And so I want to thank the nominator, whoever you are, and for the selection committee for choosing me for this award. And so today what I want to talk about is some of the work that my group has been doing for about the past 10 years or so. And so I'm not going to dive into too much in any of the nitty-gritty details of anything, but kind of give more of a little bit of an overview of what we've been doing, looking at reactive oxygen species in marine systems. And I'm going to start off kind of diving a little bit into the ecology of that system, but then bring it back out into why we think it's important in marine systems. And then in the spirit of this lecture, in terms of talking about approaches and innovation, I'll talk about some of the new instruments that we've been developing to try to start making significant advances and discoveries in this field. So to get us started and oriented, I know not everybody thinks about ROS on a daily basis, but reactive oxygen species, or ROS or ROS, I'm going to use those all kind of interchangeably, are all oxygen intermediates that form during the sequential reduction of molecular oxygen to water. And so there are a number of different oxygen intermediates, or ROS. I have some of them listed here. The one I'm going to mostly focus on today is superoxide. Superoxide forms when you donate one electron to molecular oxygen. And it's highly reactive, as the name implies, and it can undergo a number of different reactions, both oxidation and reduction reactions, depending on what it's reacting with. It has a short lifetime in most systems, on the order of about 30 seconds or so. And as the name implies, again, it is super reactive. And because of that, it's believed to induce a lot of negative consequences to biological systems. And in fact, many of you probably, when I first mentioned ROS, or when you saw reactive oxygen species in this topic, the first thing that probably came to mind was stress, or oxidative stress, or oxidative damage. And that's what most people think about when they think about ROS. And this is a great article that I found that was published in the New York Times in the early 90s, The Price We Pay for Breathing, which I just love the overly melodramatic nature of this article. But they go on to say, in the age of modern triumphant, all things radical bear a bad connotation, but surely nothing can match the ill repute of free radicals. And that really does actually ring true, that free radicals, if you Google that, you're going to get all kinds of crazy images of, devils with spikes, and, free radicals really do have a bad reputation. And, and there's some reason for that. There's justification for that. ROS, in particular superoxide and hydrogen peroxide, have been linked to a number of different diseases and disorders within the human body, for instance, and this is just an illustration to kind of illustrate, to show some of those disorders. These range all the way from cancer to premature aging to diabetes. And so there's a justification for why we have this negative connotation associated with ROS. However, if you go on to read this article a little farther, you'll see it says, yet, like so many of society's maligned, free radicals are often misunderstood. And this is something that I kind of want to talk a little bit about today, and what I'm hoping that you leave here today is with that understanding, that there are actually a lot of beneficial properties of reactive oxygen species, not only within ecology and within organisms, but also within the ocean. And so, just to kind of orient you, when we think about reactive oxygen species, we have to think about where they're being formed. And so this is my really horrible depiction of a bacterial cell, where you kind of have the intracellular environment, you have your extracellular environment. Now, when we think about oxidative stress or the negative consequences of reactive oxygen, we're typically talking about the intracellular formation of these oxygen radicals. And so this is forming in this intracellular environment. So I have superoxide here that's being formed intracellularly. This typically forms as a function of respiration or photosynthesis. Any organism that's living within an oxygen environment are going to naturally just form ROS as a function, a natural byproduct of breathing and respiring and photosynthesizing. Now, those concentrations are beneficial. It's only when they get elevated that all of a sudden we have oxidative stress, damage to proteins, damage to the lipid bilayers, and so on. Now, superoxide can dismutate and form hydrogen peroxide, another reactive oxygen species. Hydrogen peroxide can diffuse in and out of the cell, either freely or it can be transported out, whereas superoxide cannot. And only in really highly compromised cells is it believed that superoxide will actually be transported or diffuse out of the cell. But superoxide undergoes acid-base chemistry, so it can actually protonate, and this protonated form can actually diffuse out of the cell. However, under most environmental conditions, most physiological conditions, the pKa of this reaction is 4.8. So the amount of superoxide that's going to be protonated is really small. It's less than 1%. So the amount of superoxide that's going to be protonated is a small amount, so the amount that's actually going to diffuse out is a very, very small amount. So it's believed that if you measure superoxide outside the cell, that means it was formed outside the cell. And in fact, we know that organisms also form reactive oxygen species, not only superoxide, but also hydrogen peroxide outside the cell, intentionally, purposely, using different processes, and these have very diverse functional roles. And in fact, this has been really widely studied within higher eukaryotes, including fungi, plants, animals, and mammalian systems in particular. This has been worked out for many, many decades. And there's a suite of enzymes, or NADPH oxidases, that have been studied and really well described. This is a family of enzymes called NOx that are known to do this process. And I'm not going to dive into all the details of that, but just to tell you that these are transmembrane enzymes. So they cross the outer membrane, and they transfer reducing equivalents from inside the cell outside the cell. So they transfer electrons out. So what they do is they take NADPH from the inside of the cell and they transfer those electrons outside, reducing oxygen on the outside to superoxide. Now, it's been shown that that process is really highly regulated and involved in a number of different physiological processes, including cell signaling, cell growth, cell differentiation. For instance, when fungi want to divide their hyphae, or mushroom wants to form a fruiting body, that's all formed by extracellular superoxide. Cell defense, your immune system, for instance, this is extracellular superoxide, and wound repair by plants. So these are all done by this extracellular superoxide process that's really highly regulated. And this is also known to occur in pathogenic bacteria as well. But when we think about, so what we know for these organisms, and so what we know for in eukaryotic systems and within pathogenic bacteria and strains as well, what we have is this Goldilocks scenario. And so concentrations of ROS have to kind of hit in that sweet spot. If they're too low, then organisms can't undergo the essential physiological functions that they need to grow. They can't undergo cell signaling, cell growth, differentiation, and these things that are needed for your body to divide cells and so on. If it's too high, then oxidative stress ensues, and that can lead to cell death and all of the diseases that we talked about previously. So you really do have to be sitting there right in the middle. It has to be that sweet spot. But when we talk about marine systems, and actually I should say in environmental systems in general, that we really don't think about those beneficial roles of reactive oxygen species. Instead, we really just kind of focus on this. So when we think about the ecology of marine microorganisms, it really focuses on oxidative stress, lipid peroxidation, damage to proteins, DNA damage, cell death, coral bleaching, red tides, harmful algal blooms. These are all the things that are associated with high concentrations of reactive oxygen species. And we know very little actually about how ROS are involved in the ecology of marine microorganisms or environmental microorganisms in general, and how that is involved in controlling population structure and controlling the activity of microbes within the environment. And so I became very interested in this early on, and two of the studies that kind of got me really interested in this came out in the early 2000s because they showed that two very common marine phytoplankton, Thalassia syra and Coccus, both produce extracellular superoxide outside the cell in happy, actively growing populations. These are not cells that are stressed. They're growing in ideal growth conditions within the lab. And so I know this is like a weird unit, attomole per cell per hour, but if you actually think about that on a per cell basis, they're actually making a significant amount of superoxide, and this actually can make can have a big influence on marine superoxide levels. And so, this got me thinking that, well, maybe there's a broader role for superoxide production beyond just these two phytoplankton within marine systems and within marine microbiology in general. And so I'm going to just, before I go into that a little bit more, I'm going to tell you a little bit about the approach on how we make these measurements. So for making extracellular superoxide measurements in marine organisms, we use a commercially available instrument called an FE-Loom or a Phi-Loom, depending on who you talk to. This is an instrument that was developed and is currently still being made by Whitney King at Colby College. And it's really based on a very simple premise. So it's a flow injection system. You flow water into the instrument. It mixes in a high precision mixing cell with a chemiluminescent probe. This chemiluminescent probe is very specific to superoxide, and it gives off chemical light. And that chemical light is measured in real time by a photomultiplier tube. And we document that on a computer and watch that in real time. Now and this is in an enclosed box, a dark box, because you can't have any incident light. Now for the cells, what we do is we have an inline flow chamber here so that we can introduce cells into the chamber, and then we can flow seawater, aged, filtered, sterile seawater past the cells and right into the instrument. So the tricky part here is, as I mentioned earlier, that superoxide is really reactive, right? It has a lifetime in these systems of less than 30 seconds. So you have 30 seconds to get it into the instrument and make the measurement before it's gone. And so what we typically have is a flight path of about 10 seconds before it's being measured, and then we can back calculate and model how much decay happened in the tubing during those 10 seconds. We can also spike superoxide into this aged filtered seawater so that we can then look at how much decay happens, so we can actually get decay rates of how much superoxide happened or those cells are doing when they're in this system. So we can get steady state superoxide concentrations, we can get decay rates, and then by knowing the flow rate within the system, we can get production rates. And we turn this luminescence into concentrations by running standards within the lab. So over the years, my lab has been essentially measuring everything we could get our hands on. So within our lab, we've been measuring the organisms that we've cultured over the years. Hui also has a rather large culture collection. And I've been calling up all my friends saying, hey, send me your favorite organism, and we're going to run it on the FE-Loom, and we're going to see if it makes superoxide. And what we do is this type of approach. So what you're looking at is a raw chemiluminescent trace here where you have the lumens as a function of time. So this is what a typical signal would look like. We have our aged filtered seawater as our baseline. What we do is stop the pump. We inject cells into that flow chamber, and then we wait until we hit a steady state signal. Then we'll stop the pump, add more cells, wait until we get a steady state, and so on and so forth. And then so we can keep doing that and we can keep increasing the number of cells within that flow chamber. By then knowing the amount of cells that we have there, knowing the flow rate, we can turn those into cell normalized production rates. And so then what we have is a number of different signals. So we can, knowing the baseline here, we have a signal for the first number of cells, and then the second, and then the third. And we do have a little bit of probe auto-oxidation that we have to take into account and subtract from all of our data. And again, by doing that, what we've done over the years is measured a number of different organisms that have encompassed a wide range of taxonomies, a wide range of functional groups. And so what I'm giving you here is a compilation of those organisms, and by no means is this exhaustive. This is changing all the time. This is the latest one that I've compiled. But what you're looking at here are superoxide production rates as a function of different groups and the different colors. I need to point out that these are dark production rates, so all these are done completely in the dark. So even the phytoplankton, it's dark, and phytoplankton always make more extracellular superoxide in the light, but we now know that's a totally different process. There's two processes going on at the same time, and today I'm not going to talk about the light-driven process, I'm just going to talk about the dark. And so so these are all dark production rates. And these are cells that were grown up to mid-exponential phase, so they're kind of in their happy place, they're actively dividing, they're not nutrient limited yet, they're right where they want to be, and they're growing in an ideal growth media for that particular organism. These span a wide diversity of organisms. So we have organisms that cover some of the major organismal groups within the ocean. For instance, we have SAR-11 represented. We have Prochlorococcus, Synechococcus, Thalassiocyra, Emiliana huxleyi, and so on. So all of the major groups that we could get our hands on, we have represented. One of the things that you want to look at here, this is a log scale, so you can see even though everybody that we've tested makes extracellular superoxide, the rates are very different, right? They scale several orders of magnitude depending on the organism. And I got to draw your attention down to these four down here, these small blue bars here. Interestingly, those are Prochlorococcus, one of the most dominant cyanobacteria in the ocean. Prochlorococcus is always an outlier in everything that we've done so far, and I don't have time to talk about it all today, but I'd be happy to talk more if anybody's interested. But so we see, a wide variability. Now, if we normalize these to surface area, this converges a little bit, but not completely. So there is a lot of differences within these different microbial groups. But one of the things that kept popping out at us, and now that we know they're all making it, we want to get to why, right? Why are they doing this crazy process of making superoxide outside the cell? One of the things that kept popping out with all of these organisms, we were seeing this relationship, and I have three representatives up here, Synechococcus, Phaeocystis, and Thalassiocyra, and you're again looking at production rate normalized per cell as a function of cell number. So these are the total cells in each flow chamber. And what you can see is that as you increase cell number, you get less and less superoxide production on a per cell basis. Now, for those of you who study microbial physiology, this screams of cell signaling, right? Quorum sensing behavior. At higher cell densities, you get less production of that molecule. And so we found that was really interesting. And so I'm going to dive into two organisms a little bit deeper. I have a love for the Rosiobacter clade, so I'm going to dive a little bit more into Rosiobacter. These are two different species, Rugeria pomeri and Rosiobacter azwk3b, that are both within the Rosiobacter clade, a widespread clade within marine systems. And as you can see, both of these have that inverse relationship between cell number and extracellular superoxide production rate. So why is that? Is this a cell signal similar to other eukaryotic systems? Well, one of the things that kind of was bothering me a little bit about our approach with this is that what we're doing is we're stopping the pump, we're adding more cells, we're starting the pump, we're stopping the pump, we're adding more cells. I started to get a little nervous that maybe we're stressing the cells out in this process. Maybe what we need to do is try a different approach. So what we did next was said, okay, let's take these two organisms now, let's grow them up to mid-exponential phase, and let's dilute them out. So rather than continuing to add cells, now let's dilute them out and see how superoxide production changes. Well, when we do that, we get the same exact thing. So that was reassuring. So what you're looking at here is steady state superoxide on the left. And this is as your more diluted cells as you go to the right. So this is undiluted to diluted. And as you can see, as you would expect, when there's more cells, you have more superoxide. And as you get more diluted, there's less superoxide. But again, if you convert that into a rate and put that on a per cell basis, so now on the right, looking at superoxide production rate on a per cell basis, the cells that were diluted produced way more superoxide on a per cell basis than those that were undiluted. So this, again, it kind of screams to this idea that it's cell signaling. And not only that, but they respond incredibly fast. This happens within minutes, that we can dilute these cells out, we can put them on the FE loom, and they've already responded to their environment and started upregulating this superoxide production process. And so what I wanted to do next was say, okay, well, I want to take one of these organisms. So this is telling us that this is a highly regulated process. I want to look at superoxide production over a life cycle of this organism. So what you're looking at is Reeburgh DSS3, this common Roseobacter species, over a life cycle. So first you're looking at superoxide concentration over time, and the blue dots are the optical density, so it's growth. And so we have our lag phase all the way up to stationary phase. And this is just the total superoxide concentration. So you can see as the cells are growing and they're dividing, we're getting more and more cells within the beaker, that there's more and more superoxide. The really cool thing here is as soon as the cells enter stationary phase, superoxide's gone. So this was reassuring to us for two reasons. One, this really indicates that this is a linked to growth and that this is a regulated process. And number two, this told us that this was not cell lysis. Over the last decade, I've always had a couple critics that have come up to us and said, well, how do you know that you're actually measuring extracellular superoxide? How do you know that you're not just lysing cells and that this is just superoxide oozing out of the cells? Well, this is clearly not lysed cells because this is when you would see cell lysis, is when organisms are going into stationary phase. So that was kind of a little bit more vindication on our part that what we're measuring is true extracellular production and not some artifact of cell lysis. So next what we have is we wanted to say, okay, well, clearly this is a regulated process, but organisms can regulate two ways. They can change their production, they can also change their decay. So what we wanted to see is how is decay changing over a life cycle. So we did those spiked superoxide experiments, which we had to make sure that we were adding exactly the same amount of superoxide to each of those because that's also concentration dependent. But we have our superoxide decay constant as a function of time. And what you can see is early on in lag phase and exponential phase, there's really low decay. And it fluctuates around a little bit, but there's low decay, so Roseobacter apparently wants that superoxide around. As soon as Roseobacter enters into stationary phase, though, decay rates go through the roof. And in fact, those little bars on the top, or those little lines on the top of the bars are there to remind me to tell you that those aren't actual numbers. The decay rates were actually so fast that I couldn't actually measure it. And I was spiking in so much superoxide that I couldn't get anything to come through the other side. And I eventually stopped because it was getting to a concentration that was unrealistic. So obviously, Roseobacter does not want superoxide around when it's entering stationary phase. And in fact, what you do when you look at the total superoxide concentration on a per cell basis, what it ends up being is that the concentration of superoxide around the cell is actually extremely consistent all the way through growth. So it's like Roseobacter wants to keep the concentration around the cell exactly the same all the way through growth until it enters stationary phase, and then it wants it to be gone. And so, this tells us that it's doing some sort of process, right? It's benefiting the organism in some way that it's that highly regulated. The production and the decay is so highly regulated to keep the concentration that consistent all the way through growth. And I should mention that this isn't the first time this has been seen, okay? So I have to be honest. And of course, it was in E. coli. Everything's been done in E. coli. So anytime I think I've done something novel, I look in the literature and it's been done in E. coli. It's always depressing. But in E. coli, of course, this was a pathogenic strain of E. coli. It was shown that there was, they've actually also figured out the enzyme, the gene that's involved in extracellular superoxide production. If they upregulate extracellular superoxide production, E. coli grows to these mad densities, extremely high cell densities. By knocking out the gene that makes extracellular superoxide production, E. coli couldn't divide, and it formed these really kind of morphed cells where it couldn't actually divide. So I think there's something similar going on here. Unfortunately, I don't have the enzyme figured out for this organism. I do for the other Erosiobacter, but unfortunately has 3 regions of the genome that encode for the enzyme. And if we knock out all 3, the organism can't live. So there's no way of actually knocking out the enzyme and then seeing how that impacts the growth in Erosiobacter. But we can just scrub the extracellular superoxide, right? So we can just get rid of it and see how that impacts growth. So there's an enzyme, superoxide dismutase, that that specifically degrades superoxide. And so we added that exogenously to both of those Erosiobacter strains and said, okay, how's that going to impact growth? So you have 2 growth curves here where you have without superoxide dismutase added, and then added different concentrations to see how it impacted growth. And just like we would expect, what happens with E. coli is we get a near complete inhibition of growth when you add superoxide dismutase. So we're really excited about that. Unfortunately, I can't tell you why. We can't tell you why this is happening. What is exactly a superoxide doing on a mechanistic level? We don't know. In E. coli, it's been suggested that maybe superoxide is reacting with the outer membrane and changing the biophysics of the outer lipid membrane, so allowing for it to essentially divide. It's also been suggested that maybe superoxide dismutates to hydrogen peroxide, which then comes into the cell and acts as a cell messenger. But no one really knows, and so we don't know exactly why it's involved in growth. But clearly, there's a process here where superoxide is essential for the growth and division of Erosiobacter, and likely other marine organisms. But I want to start stepping back and getting away from the ecology of Erosiobacter here and bring this out into the ocean a little bit and saying, okay, so we know that extracellular ROS is regulated by marine microbes. We know that it's widespread. We also know now that it has a role in cellular physiology. And I'm not going to talk about it today, but we've also seen similar results for phytoplankton, for Thalassiocyra. And so what is the role in marine ROS fluxes in general? So how much of a role do microbes have in oceanic ROS? And so I have to kind of reorient you if you're not familiar with ROS in marine systems. And so what do we know about ROS in the marine environment? Well, ROS has been studied for a really long time in marine systems. And primarily, it's been known to be important within the photic zone. So in the sunlit surface ocean, it's been known that ROS are important mediators of a lot of abiotic redox transformations. So the cycling of copper, the cycling of iron, and so on. But all of this was fundamentally constrained by light because it was believed and is believed that most ROS is formed due to the photoexcitation of color-dissolved organic matter. And so this is fundamentally constrained by light. We also know that from the early 2000s that with the discovery that phytoplankton were also producing extracellular superoxide, that they could be also contributing to this flux of ROS. But they're also fundamentally constrained by light, their distribution. And so, the question was, well, now that we know that this goes beyond phytoplankton and it goes beyond extracellular superoxide production, goes beyond just that organisms that are producing it as a function of light, this opens us up that extracellular superoxide could impact the dark ocean, right? 90% of our global habitat is actually in the dark. And so how much of that contributes to global ROS fluxes? And so this data points that microbes could be a likely source of ROS within these systems. And so what do we know so far? Well, unfortunately, we don't know a whole lot. And that's because it's really difficult to measure reactive oxygen species within the environment. And most of what we know is on hydrogen peroxide because it has a lifetime of several hours. So it's a lot easier to measure in that context. However, there's some evidence that it's actually a little bit faster than we believed in some systems now. But for superoxide, you have a lifetime of less than a minute. So for traditional oceanographic methods, you throw your CT2 over, you bring it back up on deck, you have your Niskin bottles, you bring those into the lab, you hook it up to the FE loom. By the time you've done that, superoxide has cycled over so many times that you no longer have it present within the system. So what have we done in the past, and what is the current methodology? Well, there's two main ways that people have made measurements of reactive oxygen species like superoxide currently. And one of them is that you take those Niskin bottles, you bring them in the lab, and you take water, and you have them in a dark bottle. You incubate them at that in situ temperature, so you try to keep them similar to what it would be like within that packet of water within the ocean. And then you hook it up to the FE loom and you measure steady state dark concentrations of superoxide, okay? So that's not mimicking the environment completely, but it's at least giving you some sort of idea of those dark concentrations. The other way is that you take that water and you filter it, and you monitor and you model the decay. And by modeling the decay of that water, then we can project back to the time when the water was collected, by the time when the water was filtered. We can project back what was the in situ concentration, and then by knowing the decay rate, we can get production rate. But that only gives you particle-associated superoxide production, right? So those are really what we have. We have dark particle-associated superoxide production rates. But that's the best that we've got so far, right? But by doing that, it's been shown by the handful of studies that have been done that dark particle-associated superoxide is being produced not only within the photic zone, but all throughout the water column. And there's been a number of groups that have studied this over the years, and I'm showing you some rates from two different groups, from David Waite's group and Bettina Volker's group, giving you some indication of what those rates look like. And then on the right-hand side here is a study from Andrew Rose, showing you that below the chlorophyll max here, we have superoxide being produced all the way down into these deep, dark waters. And so we do have extracellular or superoxide being produced. That's particle-associated, so you get those essentially by doing those filtering approaches. Now, my group has also been trying to expand on kind of our breadth of measurements using this same approach. So we've gone to a number of different systems and tried to look for these dark particle-associated superoxide concentrations. And so here's some data that we got from a cruise a few years ago along the eastern seaboard. We hit a number of stations along the east coast in different phytoplankton blooms, and then one station that was off-shelf. And then, so you're looking at the chlorophyll max here, and then the temperature, and the steady state superoxide concentration. Again, this is dark superoxide. And the gray here is just to indicate you're now here in the aphotic zone, and this is you're in the photic. So we have dark production of superoxide all throughout the water column, including the photic and the aphotic zone. But the interesting thing that came out of this was we said, okay, well, let's filter some of that water. Let's see how much of this steady state concentrations of superoxide are associated with particles and how many are not. And so by filtering, what we found was that it varied quite a bit from station to station. And so on the left-hand side, you're looking at coastal Maryland. By filtering those waters, we pretty much removed all sources of superoxide. And so it was pretty much all particle associated, likely microbial, but we can't say that. However, in coastal New Jersey, it was not the same. So there was also a production of superoxide that had nothing to do with particles. So this is likely reduced metals or reduced DOC that's giving us these concentrations of superoxide. Of course, we can't say that it wasn't small organisms that eked through the 0.2 micron filter, so picoplankton that also got through through the filters. But likely also some abiotic contribution there. Could also be extracellular enzymes, of course. In our off station, these non-particle associated superoxide contributions are a much bigger factor. And what was really interesting is we're getting down to 2,000 meters, and we still have superoxide being produced, and it's not particle associated. So there's a lot of superoxide being generated by processes that we just don't understand. And it's likely abiotic in nature. It's likely related to some sort of reduced metals. But to get back to this, we just really don't have a good, that's as good as we've got right now. So that doesn't take into account photochemistry. That's really only kind of getting at this, what is particle associated, what is not particle associated. So we have to do better, right? So we have to get a better understanding of ROS in the ocean. And so to do that, we have to get away from using these types of traditional methods. And I want to point out also that, it's not just microbes that can be sources of ROS. We also believe that there's a lot of other sources of reactive oxygen to the ocean that aren't microbes. And these include corals, these include sponges, kelp, seagrass, for instance. And these aren't organisms that we're just going to pull up into our niskins and bring on deck, right? So we need to be able to make these measurements in situ. And so, and I should say that in thinking about corals, this is kind of a timely thing to be looking at. So corals have been bleaching, as many of you know, worldwide. This is a global catastrophe where corals are bleaching and dying, and ROS have been accused of being the main culprit of that process. However, we don't have any measurements of ROS in corals. And so to be able to start making these measurements, to start actually looking at whether or not there's a relationship between stress in corals and ROS is kind of somewhere we need to go. And so my group over the last few years has really been trying to make some strides in this area, and I'm very fortunate to be at WHOI, where we have some wonderful engineers who I've roped into this project and convinced them that superoxide is someplace that they want to study and spend their time. And we've got a whole series of different sensing technologies that we're starting to develop that I really want to just completely open up the ROS landscape within the ocean. And this ranges all the way from the cellular level, so from the polyp scale, like microsensors, all the way up to the ecosystem scale, so reef scale, or just large-scale ocean basins. Can we actually understand the full ROS landscape within these systems? So today I want to just introduce two of those technologies to you, so that are the furthest along, and so you can see where we're at with those. And the first one is my favorite because I do love DISCO, and it took me a really long time to come up with this acronym, but it was worth it. So DISCO is the Diver-Operated Submersible Chemiluminescence Sensor. And DISCO was it was funded by Schmidt Marine Technology Partners, and they were the first ones who gave us some funds and said, we're going to have faith in you that you can make this instrument and do this in situ. And so it's a handheld instrument that's rated to 30 meters. And it's really based on the same exact technology as that FE-Lum. So it's really just a flow injection system where it uses that chemiluminescent probe. It's a photomultiplier tube. The tricky part is that it all has to be submersible. It has to be able to withstand pressures. And everything has to be inert, right? And so what we're finding is that most of the products that you find on the market are not inert. Even if they tell you they are, they're lying. Nothing's inert, at least they're not superoxide inert. So even rubbers have zinc in them, stop valves have really tiny amounts of stainless steel in them. And so we've ended up having to manufacture most of everything, all of the parts in-house. And so this is the DISCO here, where we have a little tablet that we can actually control the voltage on the photomultiplier tube. We can change the flow rate. We can look at the concentrations in real time. This is a picture of my student, Kalina, measuring superoxide. And you can go up to the coral, you can measure superoxide, you can take it away, measure the background seawater, go back over to the coral, measure superoxide. And so what a typical, so we deployed DISCO for the first time. We were lucky to get a cruise with a colleague at WHOI to Gardens of the Queen, nice place to go for the first time with DISCO, and got to dive on these beautiful, pristine coral reefs in Cuba. And just an amazing reef ecosystem, very healthy corals. And so this is what a typical trace looks like. And so what you're looking at is the raw signal over here versus time. This is us kind of swimming around. This is our background seawater. And we would go up to a coral, say Orbicella faviolata, and hold the wand there and see nothing. And it was like, uh-oh, the sensor's not working. This is not good. You know, back to the background seawater, back to Orbicella, we get nothing. You know, so there was a lot of sweating and concern at first, but then we go over to Porites, and the sensor goes through the roof, right? So we learned really early on that there's a really big, wide species diversity, a specific signal in these superoxide levels. And so you can see that, you know, we go to, the colors are the different corals, and so the Porites species would have these big signals, and then other species would not. Sometimes we'd get this hysteresis effect where we wouldn't go all the way back down to background levels, which we believe, you know, corals make a little bit of mucus. They have mucus on the outside of their colonies, and we think that gets a little bit of carbon stuck inside the flow sensor. So we're currently working on a way of doing like an acid rinse in between. So instead, what we do is we use this new background when we normalize all of our data for the time being. Then we can inject SOD to confirm that what we are in fact looking at is superoxide. By going to these different reef sites, then we can start compiling all this data, and we are seeing all these trends kind of start popping out. Porites species make a ton of superoxide, some species make none. Unfortunately, some species we only got to sample one colony. So this is an ongoing project. My student Kalina, this is her main thesis project, and we're going to be doing more dives in the near future here to really try to understand what these species level variability means and why are these corals doing it. These are healthy corals. This has nothing to do with any sort of stress signal. So the other, you know, we wanted to take DISCO further. So this is limited to 30 meters. This is limited to, you know, it's not limited to corals. It can go to other ecosystems like, you know, seagrass and so on. But we wanted to be able to go deeper. So this is Solaris. This is the instrument that we just finished this year. And Solaris is essentially the DISCO on steroids. So it can go down to 4,000 meters. The tricky thing here is that because we're going down to 4,000 meters, we have to be able to go across a really steep temperature and redox climb. So we have to be able to do our calibrations in situ. And that's because, you know, the whole basis of this measurement is a reaction. It's a reaction between a chemiluminescent probe and superoxide, and that's going to be temperature dependent. So we have to be able to do these calibrations in situ, and so Solaris has the ability to do calibrations. It can make superoxide and make a calibration in situ. And so Solaris was just recently tested in October on a cruise out in the Monterey Bay and the Davidson Seamount and Channel Islands. We were out on the submarine Alvin, where Solaris can integrate into vehicle platforms, either HOVs or ROVs. And then it can also integrate into a rosette. So we also integrated it onto a CTD platform. And so, unfortunately, this data has not been processed at all yet. We just recently got back, and so, but I wanted to be able to show you at least something from the cruise. So this is completely raw data, and I don't have any of the CTD data processed, but this, I wanted to show you at least one of the corals. So this is Swiftia. This was down at 1,300 meters, so a really deep coral colony. And here's the Alvin manipulator holding the wand up to the coral colony. So here's the seawater, here's colony number 1. We go back to the seawater, the manipulator holds it back to the second colony over here, and you can see the signal, and then we would inject SOD to show that we were in fact measuring superoxide. So it works, which was surprising. This was the first prototype. We were fully convinced that it wasn't going to work, that it was going to leak or implode or whatever was going to happen, but it worked, and we got measurements, and this data is currently being analyzed, and stay tuned. We'll have lots of that to show soon. So, you know, to kind of wrap this up, you know, what we have are seas of superoxide, and Yala Shaquette and Andrew Rose came up with this saying in a perspective they did, a perspective paper, where we really do now know that we have superoxide being produced everywhere in the ocean, and it's not just limited to sunlit areas. And this isn't limited to superoxide either. It's hydrogen peroxide, hydroxyl radical, which is, many would argue, is the most reactive of the reactive oxygen species, and going to have a big consequence not only on the ecology of the ecosystems, but on the biogeochemistry. And I should say that I got into all of this by studying manganese. You know, my background is more as a mineralogist, and I studied, I was in my postdoc and I was studying this Rosiobacter species that I talked about earlier. And I had isolated it in Elkhorn Slough next to Monterey Bay. And this Rosiobacter species oxidized manganese to manganese oxides. And when I was trying to figure out how it was oxidizing manganese, I discovered that it was producing extracellular superoxide. And that was the beginning of the end for me in terms of my whole group now is obsessed with superoxide. And we showed that it was this concentration-dependent inhibition of manganese oxidation as a function of scrubbing superoxide. But since that time, it's been shown by my group and others that this extracellular superoxide production process is important for manganese oxidation well beyond just my Rosiobacter. And it's been shown in a number of different fungal species. We have a paper that just came out showing this is also used by phototrophs to make manganese oxides. And even the strains that Brad Thibault has been studying for many years that oxidizes manganese using a direct enzymatic pathway. Secondary to that, they're also oxidizing manganese using superoxide. So there's two pathways going on at the same time. And so there's a lot of complexity to this process and the role of superoxide in that. And so, what we've shown is that, there's this intricate link between microbial superoxide production and the manganese cycle. And what we ended up showing was that not only do the microbes need to produce superoxide, they have to get rid of hydrogen peroxide to be able to make manganese oxides. And the reason for that is that hydrogen peroxide is also really reactive, right? So I focused all of today on superoxide, but hydrogen peroxide itself is a very reactive species, and it can react with a lot of metals, and it's a fantastic reductant of manganese 3. And so if you don't get rid of the manganese, of the hydrogen peroxide, then you end up actually just reducing the manganese 3 back to manganese 2. And so you end up getting this kind of cycle of between the reactive oxygen species and manganese species where they're just cycling around. And there's a lot of interest in this right now because, there's some ocean basins, for instance, that Peter Kroot has shown, for instance, where manganese may be scrubbing reactive oxygen species just on catalytic amounts of manganese. You only need a tiny amount of manganese to just scrub all the ROS out of that system. And there's also a lot of interest in thinking about this for the origins of life. And was manganese the primordial antioxidant of the ocean? So there's a lot of really fun things to be thinking about in terms of metals and manganese and the link with the reactive oxygen species. And it goes well beyond manganese. And so this does not do justice to the role of reactive oxygen in terms of its role in other elemental cycles. But we know that superoxide reduces iron. It's actually been suggested that this is maybe one of the reasons why microbes make extracellular superoxide. Maybe it's to reduce iron 3 to iron 2 so they can take it up. And this has been shown for some species, including trichodesmium. It can also reduce mercury and lead to evasion. And then superoxide will go to hydrogen peroxide, which, as I said, can induce a lot of different reactions. And then we'll get the formation of hydroxyl radical, which is one of the main pathways for carbon remineralization within the environment. And so there's a lot of room to explore. And as I said in the beginning, a lot is known about reactive oxygen species, the reaction kinetics between ROS and metals, but now we know that there's just so much more landscape of that within the ocean, and where all of these crossings of these cycles and how microbes are mediating this is really just starting to get looked at and is a new frontier, in my opinion. So with that, I'd like to thank my postdocs and students. I have been extremely lucky to have an amazing group of people in my lab, and all of the work I'm showing is them, not me, and collaborators at WHOI and beyond, and then funding agencies who believed in us to do this work. And hopefully there's time for questions. Thank you.
Wow, thank you. What an exciting talk. We'd like to take a few questions. We do have some time. And there's some microphones.
Hi, Colleen. Great talk.
Thank you.
I thought it was very interesting to see that the superoxides produced in both the shallow coral reef and in the deep Swiftia. So presumably that deep Swiftia is in the absence of light. So
Right.
it wouldn't have photosynthetic zooxanthellae. So that points to possibly it's being produced either by the coral itself or by its microbiome. Do you have plans to try to separate those two so you can tease that out?
That's a great question. Yeah, I didn't really dive into all the coral aspect, but yes, so we've done a little bit of work showing that it's actually decoupled from the algal symbiont. So we've done work looking at a diel cycle and showing that extracellular superoxide production is the same in the light and in the dark. We've looked at bleached corals that have low levels of symbiodinium and the concentrations are the same. And now with these deep corals, clearly there's no algal symbionts there. My student Kalina is actually really interested in trying to figure out how much of this could be bacterial symbionts versus the animals. So she's actually just recently started doing some work with coral larvae and showing that they make extracellular superoxide as well. So we're starting to try to extract the enzymes, identify what those enzymes are so that we could monitor those in situ.
Thanks.
Yep.
More questions?
Hi, Colleen. That was a great talk. I'd like to take you back to your roots and ask you do you think there's any role for the superoxide and other oxygen species in soil cycling of metals, for instance, manganese, iron, and chromium, and would this play a role, and do you know anybody that's working on it?
Yeah, great question. Yeah, so there are some groups that are interested in looking at in aquifer systems, I think in particular. There has been some work looking at mostly, I think it's abiotic, that what people have looked at is how does reduced iron, when it gets introduced or oxygenated, produce ROS, primarily hydroxyl radical during that process. But I definitely think that in soil systems, microbes are going to be making ROS and contributing as well. We've also been starting to look at mineral surfaces as sources. So mineral surfaces are also playing a role. So once you dive into the soils, it's going to get really messy really fast, as I know from my roots. But I think it's really exciting. Andrew Rose, I know, is excited about getting into that and trying to come up with ways of measuring profiles in situ, like developing kind of gel technologies to look at profiles of superoxide. But I think there's a lot of room to get to that. Yeah.
Thanks.
Mm-hmm.
Hi, yeah, I'll second that. Thank you for a wonderful talk. I just want to ask you a question as a tourist. You know, I love coral. Don't know very much about them, so I wanted to ask you about where you're going with the work on coral. To the extent is it about just learning how that very interesting, complex ecosystem works, or do you see that maybe there's going to be a stress response, say to heat or acidification, that's going to produce more of the ROS that would create more decoupling of the mutualism? So, you know, just thinking ahead to when all your disco and Solaris are going.
Yeah.
What does it mean for me, the coral lover?
Right. So, you know, I think my gut reaction is that I think that the corals are doing it just like with fungi. We know that fungi produce it for a lot of different reasons. It's like the Swiss Army knife of molecules, right? And so I kind of think that corals are probably doing the same thing. I was talking to Santiago Herrera earlier this week, who's a deep sea coral biologist, and saying that we imagine it's linked maybe in deep sea corals to carbon acquisition, right? So corals in the deep are getting this chewed up carbon as it's coming down and raining down into the deep sea, especially when you're sitting down at 1,300 meters. You don't have the good stuff, you know. So being able to break down that carbon and take it up may be a way that these corals are doing it. And if they can make superoxide and hydrogen peroxide and help, you know, break down the carbon and take it up, that may be a strategy, which we know fungi do that as well. For the shallow corals, you know, we're really intrigued by the fact that Porites, which is known to be a really weedy coral, Porites has withstood many global extinctions in the past. I mean, Porites is a really, really hardy coral, and it is producing a ton. And you know, there's a massive coral disease outbreak right now in Florida, and one it's moving into the Caribbean. And Porites is one of the species that isn't actually impacted. And so we're wondering if there's maybe an immune response there. So, you know, when we get sick, one of the first things we do is our body attacks, defends itself from pathogens by producing ROS. So maybe, you know, of course, this is me arm waving because we don't know, but maybe that with Porites, they have this fantastic immune response, right, where they can just fend off pathogens. And so we have some ideas of doing some aquaria incubations where we have a species that can't make, like Montipora is a species that doesn't seem to be making superoxide, and a Porites species that is, and having them in aquaria and introducing them to pathogens and seeing how superoxide production compares. So we have a lot of ideas and ways that we want to move forward with that, but you know, at this point it's all arm waving, but yeah. Does that answer your question? Okay.
You have time for one more? Hi, that was a really great talk, thank you. I was intrigued by the very beginning part where you're talking about the Rosiobacter work, and you showed that the Rosiobacter had constant levels of ROS around their cells. And it made me think about, actually more related to the coral stuff too, which is what happens when you have two bacteria together, or an algae and a bacteria together, and they have different levels that they're regulating to. Have you looked at co-cultures, or do you have any interest in looking at starting at even that level and then going to the community scale, but you know?
Yeah, we haven't done cold cultures yet, but I really want to. Yeah. And I'm kind of frightened to get to that because I think it's going to get really complex.
Yeah.
We did some, this was many, many years ago. I did have an undergrad in my lab for one summer, and we did some on plates where we had two fungal species interacting. We had a fungi and a bacteria interacting, and we used these ROS probes, which are, you know, there's some artifacts there, but we looked to see whether or not we could see changes in ROS production when they started to interact with each other. And what was really interesting was that some species interactions we would see more ROS production, and other species we wouldn't see any change, and other ones we would see depressed production. So there was definitely species recognition, and then there was changes sometimes in the regulation of that ROS. But we haven't done it yet since that time. But I think, especially once we get into corals and we're getting into this, you know, corals are scary because it's a zoo in there, right? It's so many different organisms and the animal itself. We're going to have to get there to actually start to tease it out. Yeah, thanks.